Today I will talk mostly about my research. The research will be related to the previous talk, and the first, I think, 15, 20 minutes I will use to quickly go through what I went in the first talk, but then you will see how this picture of positional information and information processing gets complicated for various reasons. So, yeah, as we've seen last time, when I think about organisms, I like to ask this question: how is it possible that from a single cell, a multicellular complex organism develops in a very reproducible and precise way? And here you see this single fertilized cell. The cell divides, cells quickly increase in numbers, but early on, the cells are very similar to each other. Here they were stained with fluorescent markers, so we could trace their fate. But apart from that, they would be indistinguishable. But already 10 days after fertilization, the embryo has its shape, has its form. The organs start to develop, sophisticated structures emerge. And yeah, we could appreciate this blood vessels in green, cell nuclei in blue, and actively dividing cells in red. And what is amazing here is that cells know exactly when, where, and what biological function they need to perform. And it's unclear, how do cells acquire this information? And the answer is they acquire it by reading the concentration gradients of the morphogens. So morphogens provide kind of coordinate systems for the cell. So again, quick reminder for people who haven't been here, like the morphogen is a signaling molecule that is produced at a specific region in the tissue, and then it spreads across the tissue. Here you have the tissue. In every cell there is a regulatory network interpreting the concentration of the morphogen, and depending on the level of morphogen, signaling activity, different genes are expressed. Here, if the level is high, you get blue gene. If the level is intermediate, white gene, and for low expression, you get the red gene. And this creates, as a result, a gene expression pattern that has stripes. And these stripes will later on give rise to different types of organs, body parts. In my case, in spinal cord, it will give rise to different types of neural progenitors that then will get maturated and will become neurons. And in the first talk, I was sorry. In the first talk, I gave you this framework that you treat the morphogen signal as an input. You treat the gene expression pattern as the output, and then the network acts as a decoder that takes the useful part of signal from the input signal and then results in producing the output signal. But then this picture is already in the developing embryo complicated for various reasons. One is neural tissue in the spinal cord undergoes change in size and shape over time. So here you see how it looks 8 days after fertilization, and then over 48 hours, here it starts to fold like you have initially neural plate, so it's not the tube, then it starts to form, and then it closes. So there is change in shape. And okay, when you have this tube, or even before, you already have sources of the morphogens. So again, just a reminder, what are the morphogens we are talking about? So we will take the ventral part. There is sunny hedgehog, which is produced by the floor plate. On the dorsal side, this is BMP, that is produced by the roof plate, and these morphogens spread across the tissue, form gradients of concentrations. These are read out by the cells, creating the stripe pattern of neural progenitors. There are exactly 13 domains of these different neural progenitors, and this will give rise to different neurons that make. There are some interneurons. There are some neurons that are responsible for sensing different things from environment like heat and so on. So you probably want to have this structure well established. But apart from this change in shape, there is also in the spinal cord particularly, there is growth. So things here take time compared to Drosophila, where you had this pattern emerging on the order of minutes and everything happening basically within 2 hours. So here, after the neural tube is closed, the neural tube extends fourfold from 100 micrometers to 400 micrometers in this dorsoventral extension over 60 hours. So just how this was measured. My collaborator Anna Kicheva, she took 300 spinal cord cross sections taken at different embryonic days. Then those cross sections were immunostate and to mark expression of key target genes and signaling activity of the morphogens. And now let me stop for a bit and say aloud the questions, the research questions I will be focused about in this talk. So first, how are morphogens interpreted to result in precise and reproducible patterns? So this is the signaling part. Then, how this signaling part is affected, or how the pattern formation is affected by tissue mechanical properties, cellular dynamics, and growth. And next, I will because so far I was saying, the morphogens are produced by the specific region in the tissue, but the story is more complicated because initially the source is not there. So the source of the morphogens is also established as the tissue is growing. So it's kind of part of the pattern. So I will get to that, how such a source can be established. And finally, and this will be like really brief one, I'm from Institute of Theoretical Physics, so we also do some theory here, and this will be to show you that you could solve some things analytically and then get some general insights into how stable patterns can emerge. Okay, starting with the first one. So you have seen this, different gene expression, like this fluorescence in these images. So if you quantify that, we have BMP signal, sonic hedgehog signal. These are different time points of development. And early on, and in the bottom row, you have target gene expression patterns. So what you can notice, and you could also quantify that, early on, this morphogen signaling activity, the profiles are exponentially decaying, but because this is in relative units, so you divide the position by the length of the tissue, and keeping the constant decay length of each of the morphogens, if the tissue is growing, you kind of have this morphogens moving away from each other. So this is different than, let's say, in Drosophila, we had the scaling of the signal. Here it doesn't scale. Here it's really if you would be at a specific position, it would change over time. And for the target genes, initially they are correlated with the input signal, but around 30 hours they basically form the sigmoidal shape, which basically corresponds to forming boundaries. And now we wanted to quantify how much positional error, or how well cells could interpret the morphogen signal to specify their position. And again, applying the formula that I introduced last time, but just to give you intuition, it's a kind of simple thing, because you take the spread in the gene expression, or in this case, in the morphogen signaling activity, and you transpose it to what would be the uncertainty in the x position. So if the slope of the gradient, this is okay, this whole thing is like inverse of the square root. So if that would be small, saying like a flat gradient, then this would result in a small number, meaning that if you take the inverse, the error would be large, and the vice versa. If you would have a steep gradient, this would be large, and the inverse would result in very small positional error. So, so what you can see by applying this to these input signals, that over time, the positional error gets increased in the middle. This is not a surprise. We saw that as morphogens move away from each other, there's basically very weak signal in the center. So if the embryo wants to, taking embryo perspective, specify cells precisely, probably doing it early times makes sense, and it's what you see. This 3 plus minus 3 cell diameters are early on. The signals are sufficient to provide this level of positional information. And then we took the output genes and quantified what's there in precision. So basically, think about different embryos, and you just get, let's say, at the fixed position, what's the variation of the boundary position, and that's what's plotted here. So the standard deviation of that, and we saw that it's again similar, plus minus 3 cell diameter. So this is taken at 60 hours. So this is early time point, this is late time point. And what was interesting is these numbers match. So it might be a coincidence, but it might also indicate that the cells are processing the available information close to the optimal limit of processing of noisy signals. And again, yeah, like this I introduced, so I will just quickly say how we use that. GI is the input signal. Here, G1 is BMP, G2 sonic hedgehog. They are measured at the same time for every position in the embryo. Then we also assume that the distribution of the signal at every position, we approximate it with a smooth Gaussian distribution, bivariate Gaussian distribution in this case. And this is mostly technical because one could take more measurements and work with the actual distribution, but it's just easier to work with something that, it's more reliable to fit it with smooth distribution. And this can be constructed with having the mean of the signal and the covariance matrix of the signal. And now having that, so that's the distribution of the signal at every position x. We want to know what's the posterior distribution. So we want to know, given specific concentration of the morphogen, what would be the most likely position at that cell could take. And here, position could be if this is peaked in one, if it has for every position a very sharp peak of concentration, then this can be also unambiguously related. Position can be unambiguously related to the fate. Again, this we already discussed. Here, just this is the pure distribution of implied cell fates or position, which without any signals, this is uniform. Cells don't have any idea where they are in the tissue. This is the measurement. So what is measured? This is the normalization factor. And using that, one at least under the assumptions that we had, that this is interpreted as a specific time moment, the optimal decoder contains all the information that any cellular or computational mechanism could extract from input signals. So we apply that these profiles that I showed you, the early time point. And what we see here is that the cell fate is decoded using the color map, with red corresponding to dorsal fates, white to intermediate, and blue to the ventral fates. And this black line corresponds to the concentration of the wild type. So again, we had 2 exponential decays. If you plot that in log, this creates a line. And what we could see that close to the wild type concentration, the prediction is, these dots should ideally overlap with the lines, with the boundaries between these 7 domains, and they mostly do. What you can do more? Then you could try to perturb the signal and use that map to make a prediction about the pattern, and this is what we have here in the hypomorph. The sonic hedgehog production is reduced, so you actually get smaller concentration of the sonic hedgehog compared to the wild type. And then if you read here, the ventral domain should be compressed and the intermediate should be extended. So I already showed that. So that would be the wild type, that would be the hypomorph. I would say here you see this shift here, it's less pronounced, but also there are other things happening here, like cell differentiation. Some cells already leave this tissue, but that starts like in the motor neuron starts to maturate and leave. So there are other factors involved here, but in general, it agrees. I would say qualitatively and partly quantitatively, you could say, it gives you this prediction. To be more precise, you probably need to really reconstruct the gene regulatory network. But for me, what was very interesting about this map was this region, this shaded region that I didn't explain, that was marked here, and this shaded region corresponded to bimodal distribution of cell fates. Saying that here, there's indeed one most likely fate, but as you move away from this wild type line towards high concentration of the morphogens, you get bimodal distribution, meaning that the cells would most likely take one or the other fate. And that was the prediction that we wanted to test. We tested it in chick explants that were exposed to high concentration of BMP and sonic hedgehog, and what we could see, indeed, we either have red cells corresponding to dorsal or violet corresponding to ventral fates, but no intermediate, so no green cells here. But the question remained how cells could perform this optimal decoding, and the answer was, okay, let's try to see whether we could propose a gene regulatory network that will also act in this way. So I propose network composed of three key regulators, transcription factors acting in the spinal cord, where V representing the ventral fates, I representing the intermediate, and D representing dorsal fates, and applied standard ODE approach with thermodynamical model where different strengths of interactions are denoted here by different constants, and or parameters. These parameters were later on perturbed in the screen to find solutions that satisfy different criteria, and these criteria are made to be consistent with all experimental observation that we had. Like for instance, very simple thing. So we start with 10 to the 8 sets of these parameters, 13 parameters, randomly selected from some wide range of parameters, and we require them to produce a spatial pattern composed of 3 stripes. Those are intermediate and ventral in this order, and this already boils down to 10 to the 6. Then we want to also have in the spinal cord, as the tissue grows, you have some shifts of the domains. Then we also wanted to have that. That's resulted in a further reduction, and finally this constraint on decoding map saying you want to have in this high concentration the mixture of ventral and dorsal, and this resulted in 1000 cells. So concluding this part of story, as a result, we got let's say we got this 1000 networks, and then we as an input we provided noisy input signals corresponding to the level of noise that we could observe in the experimental data. And then as a result, we saw that the networks are producing this imprecision of the boundaries in a broad range, but also in this reaching this biologically observed range of ±3 cell diameters, saying that there are probably, and we already had this discussion that maybe if one would go farther, study these networks more deep, probably one would be able to find what's the difference and what makes some networks to really result in this high precision of the pattern. And now the next part of the story is, so far the tissue that was, it was represented by the continuous variable x. But of course, already from the images that I showed you, you saw that it's not continuous. It's discrete, it's made of cells, it's neuroepithelium. So that the cells are dividing, the tissue is growing, there is some cellular dynamics related to nucleus movements. So how this affects this pattern formation? And that was our next question. And here we took a different approach. We wanted to see how much, if you recall this initial picture of early stage mouse embryo where 2 daughter cells were labeled with different fluorescent markers, this is very similar. Basically Laura, a PhD student in Anna Kicheva lab, she really did a lot of experiments where the idea was to, if you want to know more about the technique, it's here. But in principle, when the mother cell divides, the cells are either green or red. So every green and red cells, they come from a single clone. It's not like green is another clone, red is another. They come from the same mother cells. You don't see the mother cells, it got divided. And the thing is, at E8.5, these are largely dispersed. Saying the cells spread in the tissue. Whereas later on, they tend to stay close to each other. And this is like if you would take the neural tube, cut it along dorsoventral, but also cut it along anterior-posterior axis, and then you unfold. So that's what we see here. And if we quantify now this, how basically the number of fragments that result from a single clone, we got this relation where here you see the clone size and here the number of fragments. And early stages are here in green, and just to quantify that, we just take the linear fit, and we called it like fragmentation index, which was high at early stages, low at later stages. Now we wanted to understand how you could get this fragmentation as tissue is expanding, growing. To do so, we use vertex model where movement of each cell is governed by the movement of its vertices, and to define this dynamics, you need to define the energy function. And here the energy function, let's say the energy function is motivated. We heard yesterday a lot about actin, eckaterin, and other things that makes cells contractile in Richa talk. But if you think in terms of like energy function, it means that cells have their preferred area. This is this elastic term, potentially with preferred area depending on time. There is also line tension, saying like if this is the length of the edge, and this is some lambda marking the preference of cells to having either longer or smaller edges, and then the cells tend to be contractile. This is the perimeter of the cell with gamma. Higher gamma means the cells like to be more contractile. And now to get the force acting on the vertices, you take the gradient of that over position of every vertex, RA. And with the force, we now assume that this motion happening there is a type of overdamped motion. So you just take you don't have acceleration like like in Newton laws, but you just take this is the velocity of the vertex, and this is something relating to either viscosity or a drag in this environment. So having defining energy function in deterministic way, one could get to displacement of the vertices. And now we model the system. Or even before, you could do some math. And again, we heard a bit yesterday about what are it's this idea of minimizing the energy. You could do that by and calculate by setting. This basically says that all the cells are having the same this elastic constant line tension and contractility. We also set the preferred cell area here to be constant, and then there are additional parameters, so we could simplify the system, non-dimensionalize it. And then if we ask what's the state with minimal energy, here you have fluid-like tissues. So the state with the minimal energy is degenerated. There are many states, and cells would have. I will show you some image of that in several slides. But then there is a whole region where you have a solid-like tissue with hexagonal cells. And then if you increase the tension even further, what happens is that cells are collapsing because they are the elastic term cannot overcome the contractile terms. So cells are elapsing. And now taking this, so this is the classic vertex model. Okay, we added the cell division. We wanted to ask what's the fragmentation. I did the simulation, and basically, yes.
When you say fluid-like, do you mean that they're just irregular, or their shape is ever changing? Like, they don't have a definite shape, it's constantly changing as a result of.
So in this, the shape is irregular. That's one thing, but I would say to have it constantly changing. So in this classic formulation, I would say without noise, without any let's say without noise, without any cell divisions, this will just arrest. It's like a glassy transition. It just goes there. But depending on the starting conditions, it will go to a different state. But then I would say it's enough to really bring a bit of noise, and then things start to move, and then this is like a fluid-like. Okay. But thank you for that question. And then when we did, or I did the simulation, basically I saw that there is no fragmentation. It's not even close to this fragmentation index that we observe here. So of course something was missing. And then one thing that we added to increase this fragmentation, or to see, the question was what are the factors that increase this fragmentation, and I'm telling you the story that is already finished, published. So I knew what's that, but it took a bit to really figure out what are the missing parts here. So one thing is the noise, which yes, this is fragmentation of the clones that were. So okay, maybe I didn't explain it well. I'll just. Oh, okay, maybe here. We measure the number of fragments, like the fragmentation index, saying like you have one cell, the cell divides. As the cell is dividing, this creates a clone. So this is a clonal population of cells, and some cells will stay together. Then this is one fragment. If they disattach, then these are two fragments. So you can imagine that for, let's say, clone size 4, you could either have one cluster of cells, no fragments, or every cell being separately. Then it's 4 fragments. And then we quantify it with fragmentation index, and this is a measure that I use to quantify fragmentation.
So would you also require the medium to be a bit fluid-like to see fragmentation?
Sorry?
Would you also require the medium to be a bit fluid-like to see this fragmentation?
Medium? No.
So the medium, should it be...
Okay. I will come to that. Saying like the medium, like the tissue. Yeah. Already having cell division. Yeah, that's one of the parts. Saying like why you let's say don't have this fragmentation is because if you are in the solid and then things are dividing. Okay, a priori you don't know whether in the solid, when you have cell division, it might depend a bit on how you implement that, because maybe you say because you introduce every cell division is perturbation. So you can imagine that if you are in the solid, things they divide and other cells intercalate. This might be also a valid mechanism. There are works where they show in some organisms that cell division might be enough to provide fragmentation even in a kind of solid-like environment. In the fluid-like, I agree. It's that would be the natural consequence. So the question is, can you get the fluid and still be consistent with, or maybe some different mechanism of this fragmentation that would act in the solid? So thank you for that question. And I will, I will answer that in a few slides. How this emerges. So again, getting back to this, introducing noise, that was one thing. So making noise in the line tension, so making cells, making the cell rearrangements easier for the cells by decreasing the barrier that they need to overcome in order to change their neighbors through so-called T1 transitions. But also what we noticed, and this was, I would say, the key factor here, is that the cells in the tissue, the nucleus of those cells is moving during the cell cycle. Basically, when the cycle starts, it's at the apical surface. We are looking at the tissue from the apical surface and modeling the tissue from the apical surface. Then it moves down to the basal and then moves back, causing like the cells to kind of first decrease in their area and then quickly increase in the area. And I will again say how I exactly defined it later on. But just imagine that now this preferred cell area, this A0 for a given cell alpha, is no longer constant, it depends on time. So intuitively, it's like cells try to elbow their way in the tissue. And this too resulted in increased fragmentation, and here it's like here you have this fluid region, but I would say due to this noise and cell area kinetics, this fluid region slightly shifted with respect to the ventral. So this is and again I will show you a movie. This is like fluid. This is the solid. You could also do some other descriptors, like I don't know, we had target shape index. So here it would be like 3.8. Here larger. But we also had this region that you would say it's really highly contracted region, very solid, but you still have some fragmentation. So T1 transition, and okay, that's maybe a bit technical, or not technical, but if we want to measure the fragmentation, it's very useful to measure just T1 transition, because such a rearrangement of the cells basically creates a fragment. If you have two cells that are adjacent to each other and they undergo T1 transition, they are no longer a single fragment. They create two fragments, and you can see that these two are very close to each other. So often I will just measure fragmentation either directly or by discussing these T1 transitions, and also T1 transitions is something you find in the literature, and so on. And just to reference to these regions, we call those A, B, and this novel subregion C that is supposed to be a solid, but it exhibits this fragmentation. And now, how this simulation, how this tissue is growing. Here you see the subregion B. This is hexagonal, solid-like, and the different colors correspond. Every the color corresponds to a different clone, and then when the cells are dividing, you don't see. They rather sometimes they disattach, but mostly they stay together. If you now in subregion C, and maybe another difference, they are mostly hexagonal or close to hexagonal, but very similar shape. So this is like solid. This is the subregion C, where you could already see it's highly heterogeneous, and you actually have, it's not hexagonal. You have like higher gons and lower like fourgons or triangles. And now if I play, you could see this ballooning of the cells, and as a result, you see that many of the clonal populations get fragmented. And if you quantify, you really see the difference. And just for completeness, I will show you how the subregion A looks like. So here, all the cells have almost the same area, and then it's also like I would say it's from a distance you just see like things are swirling, but you could also measure effective diffusion and really see that in both regions, if you just focus on a single cell and how it moves, that it really, they are not caged in one region, but they actually spread far away. That's
And fragmentation requires less division. I don't.
Yeah, that's a great question. We got this question from one of the reviewers and really spent a lot of time on answering that. And it's there is no clear dependence, I would say, in this region C, at least, because we were focused mostly on region C. You don't see a causal relation. So division might have something to do with it, but it's not like the main cause, saying like, of course the division is perturbing the tissue, but so the answer would be some of these events are related to divisions, but many other are not. But I must say that depending also on the tissue, it might be different, because sometimes you really see like and that was also a question that sometimes after division the cells tend to disattach, but here that was not the case also in the actual tissue, because that motivated us to also try to live image cells in this neural epithelium, and to some extent Laura managed to do it. I will show that movie later on. But what I want to highlight here is that you indeed have this high cell area variability in the region C. You could also, I didn't mention that, but if you do this analysis of energy function and try to find the minimal energy solutions for this highly contractile region, then it's also there. And this is work by the group of Frank Uliher. You find that the hexagons are not the only minimal solution, or you could get better by having like the mixture of fourgons and octagons, and then triangles of 12 gons. So I would say some of that remains here, like fourgon and so on. And if we quantify that across our space of line tension and contractility, we see this is cell area variability. But the question was whether this cell area variability is responsible for the fragmentation that we see, whether this is enough. Okay, before I get to that, we also wanted to see whether in the tissue we see the same cell area variability, because if we wouldn't, then of course it's like pointing you, okay, you have the simulation, you have this nice fragmentation, but this is not the region you should be looking at. You should actually look at some different regions. So we wanted to first check whether by segmenting the tissue, we match what is produced by the vertex model, and we use different cell shape descriptions like coefficients of variation of cell area, the number, the fraction of hexagons, cell shape index that basically took some sum of that, and we found that the best match is indeed here. We also did some laser ablations. I'm not going to talk about that, but yeah, you check the details in the work. Yes. Okay. The question was whether we don't expect this cell variability to change throughout development. Yes. Excellent question as well. Like that's why we did laser ablations at different developmental stages to check, and we also checked that for different developmental stages, and we found that there is a small difference, but basically it still stays there. So there is a bit of change, but not so much. Okay, and like saying quantitatively, because sure, that was our alternative hypothesis. That, and later on I will, because the fragmentation is changing, maybe the system is moving from here to here. That could be the case. Saying like, I don't know, you have different, I don't know, something changes the adhesion levels, or something changes the level of ecaterines and so on. But yeah, as far as we, consistent with the experimental data, we found that the tissue mechanical properties stays roughly in the same region. Okay, and now the question was, what of these different factors? So I introduced noise, I introduced this solar dynamics. Which of those contribute mostly to observing this fragmentation? And also to this level of cell area variability, which is quantified here. So if you would have just noise at the low level, there is, in this subregion C, there is no fragmentation. So this noise on itself, if you would increase it even farther, then you could get some fragmentation simply because having huge noise. But with the noise that we also showed is relevant for the embryo by basically finding the optimal noise, by basically fitting what is in the noise in the embryo, in the line tension, the fluctuation of the edge lengths. We found that this works the best, and if that would be just noise, no fragmentation. There is still cell area variability, but yeah, not at the level, and this is the level that is observed in the tissue. If you just have this cellular dynamics on this interkinetic nuclear movement of going up and down, and no noise, it gives you some fragmentation. It highly increases the cell area variability. If instead of the interkinetic nuclear movement, so this kind of shape for the preferred cell area, you take the linear, then almost no fragmentation. Okay, the cell area variability is still high. But if you have a combination of both, it adds up, and it adds up non-linearly, saying, it's a combination of both that results in high fragmentation and, okay, in slightly higher cell area variability. And now, yeah, I would say I would say I would be fine with just having some intuitive understanding of what we do with the cellular dynamics. But if someone really wants to know what's inside, then that's how we define it. So this is like a piecewise linear function that basically, we check that in this paper that this works pretty well and is consistent with the experimental results. So depending on the stage of the cell cycle, you see early on it slightly goes down and then goes up. And this term, this is just increasing in the volume of the cell. So cells also, they grow in size. And this is the part that corresponds to movement of the nuclei, of the nucleus, resulting in this ballooning close to the division. And as I promised, the movie of the dividing cell, we were focused on the apical cell area. That also, if this is the division time, it really goes up sharply. So we wanted to test whether this sharp increase is indeed the feature of this cellular dynamics that causes this the higher fragmentation. And okay, this is like a physics approach. So we took some exponentials with increasing like steepness, and for very steep exponential, as the cellular dynamics would reproduce this both T1 rate and cellular variation. But one thing remained. So we didn't still explain why early on this is high fragmentation and later on at E10.5 it goes down. But already we also knew that the neural epithelium, the growth there slows down. So early on, the cells are dividing rapidly. Later on, after some 30 hours, and in the signaling part, we also have this 30 hours when the pattern was established, and this is not a coincidence. The fragmentation goes down, and you see also the fragmentation goes down because the proliferation goes down. So cells are dividing slower. So, and this is something that I, intuitively maybe is obvious, but I tested it in the simulation. So this Kp is decrease. This is the proliferation rate that we decrease roughly by a factor of 2-3. And then we also did experiment where L-mimosine was used as a cell cycle inhibitor, and we saw that in the tissue we also have this decrease of the fragmentation. So to sum up this part, I would say this observation that the fragmentation early on, when the pattern is not yet there, the cells could move around, they could maybe explore, they could maybe maybe average the signal spatially. But at some point, when they are already need to devote to their fate, the tissue slows down. So they no longer proliferate at such rate, and they basically maybe not arrested, because it's still there are some cell divisions, the thing is growing, but the fragmentation goes down, and I would say this is like a proxy for patterning precision here. So now again, changing gears, now I would ask, going into this direction of the signaling, how the morphogen source is formed. And this is like the third, and I would say, the last part will be a really short one, so stay with me for a bit longer. How the source of the morphogen is formed. So far I was giving you this diagram. Input signal information equal output signal, and the source here will be the floor plate. So the floor plate that produces sonic hedgehog. But there is more to this story. In the neural tube, early on there is no floor plate. So you could ask, so how sonic hedgehog gets there? and there is a notochord, a structure that is more ventral from the neural tube, and this is the primary source of the sonic hedgehog. And this sonic hedgehog triggers the formation of the floor plate in the neural tube. So we again proposed a network model with floor plate marker. So basically, this acts as a proxy for a gene FOXA2 or IRX later on in the floor plate formation, and this is a proxy for NKX, that genes that are here at the next adjacent neural progenitor domain, and sonic hedgehog that is produced and is diffusing. So those things are sitting, not sitting, but they are located in every cell. But sonic hedgehog that is produced by the floor plate due to the expression of this gene is just floating and spreading through diffusion. But when you think about that, it's like adding additional arrow here from the output to the input. So you have the signal feedback, and it's not obvious how this feedback will affect the formation of the source and the pattern.
Sorry.
So how is it acting as a feedback? Yeah. Like because when I think about output signal, I mean gene expression. And here the gene expression, the expression of the floor plate, produces the signal. So the sonic hedgehog. So that's depending on how much of this floor plate is there, you expect different levels of the morphogen signal, and that's why it's. It could act as a feedback. Whether it's really there, this we will see. That's something that, because we wanted to focus here. So indeed here, maybe this is misleading because this n should be here, but indeed we think in terms of modeling. So we model like our space in the tissue is just composed of two domains. So I would say, in the model, it's like floor plate and everything,
So that the target part of the tissue is just as the sonic hedgehog.
Yes.
There, that feedback is not occurring. The feedback happens only.
So I would say yes. The feedback, when I meet in the feedback, okay. I would say the feedback still might be there, because depending on how the source is formed, the morphogen signal might be different. Though, okay, maybe like spoiling a bit, but because you have this diffusion and degradation, it's actually, here this part would not be so important, saying it's very robust against how the source is formed. So you still have exponentially decaying morphogen profile, and that's no surprise because we just say diffusion and degradation. So yeah, to some extent I agree. But what is affected might be the amplitude of the signal. So if you say the amplitude matters, and it might matter, then then this feedback goes towards the target tissue. But yeah, excellent comment as well. So again, we propose a model with thermodynamical description for the expression on F and N, so floor plate and NKX, and then this is basically diffusion equation, where you could notice the diffusion term, degradation term, and production term. And then my postdoc, Richard Ho, he basically came to my group from turbulence group, and so he was very good in fluid dynamics and everything. So he wrote, okay, this was just to remind that the system is growing. So you will later on see 100 to 400 micrometers, because we simulate this system of two ODEs and PDE on a growing domain, where we also screen for the parameters, the ones that were marked there, keeping the rest to some biologically realistic values that we also later on test how they affect our outcome. And the result of the screen is that we found like over 100,000 successful solutions. And by successful solution, I mean something that follows the dynamics defined by these equations and has two domains with floor plate on the ventral side. Also, we didn't want to, so successful solution for us was something taking from 5 to 25% of the tissue. We didn't want to have solutions that would take, the floor plate would take over the whole domain. And also that is formed in biologically realistic times. And this is at 60 hours, so that's when the simulation ends. That's taken from the simulation from one of the outputs. This is the expression of n and Sonic Hedgehog that creates this profile, and from here you would have exponential decay. And here you can see spatial temporal patterns, so there is some time that the floor plate is not yet present, and the target tissue, so n, is present, and then it builds up, and already you could see that probably it scales with the system size. And this is just the distribution of the floor plate sizes. Yes. And then, okay, I'm not go.
So
Yeah.
can I just ask one?
Yeah, great.
Yeah. So, how do you select your initial parameter space like these for?
Yeah, that's okay, maybe I will answer with this, saying like for these parameters of the strength parameters, we really do broad range search. Saying like we just set some ranges of parameters that are a few orders of magnitude. For diffusion, degradation, like Hill coefficients, for Hill coefficients we just use things that are higher than 2, so to have enough nonlinearity. For the diffusion, we use values that are found in the literature. For degradation, the same or the same order of magnitude. And then afterwards, we test, whether it makes a huge difference. But for the actual strengths of the parameters, so how strong is the activation, how strong is the inhibition, we just take broad range and then basically do brute force. So okay, this was a bit more fancy than what I did in this science paper in the spiral cord here. It was more like a random walk in the space, initiated with so it's not like just one random walk, going, but we actually have several random walks starting in different places, doing it in parallel. So we're quite convinced that we don't miss at least some large part of the space. Of course, it's never, it's numerics, so you are never guaranteed.
So just as a follow-up, so it says that the number of accepted parameters was quite high. I mean, 170 out of.
Yeah. So that's, I would say, because it wasn't like randomly selecting the space. It was moving.
It was targeted.
Yeah, it was targeted in the sense that you go in the space, and if you end up in a successful solution, then you continue with some random perturbation. If you end up in unsuccessful solution, then you take the step back. So, but again, that if there would be like some disconnected parts of the space that could lead to not exploring the whole space. So that's why we also started this a few times. But yeah. Okay, so that's basically to show you that there was no clear dependence of the floor plate size on specific parameter values. But what was interesting is now you take a specific parameter set and you do the sensitivity analysis. So you kind of perturb each of these parameters one by one, and what you see is that the floor plate is indeed affected. So if you increase the uniform activation, it gets higher. I don't know if you increase this guy, so how much Sonic Hedgehog affects the floor plate, it also gets higher. If you increase the inhibition, it gets lower. Sometimes it's very steep response of the floor plate size to the perturbation. But there was one parameter, basically the production of the Sonic Hedgehog by the floor plate, that very weakly affected our solution. So we thought, okay, let's perturb this to either set this to 0 or what happens if you set this to 0 to your solutions? And then we were surprised because you remove the link from the floor plate to the Sonic Hedgehog, saying like, now you have the source of the Sonic Hedgehog that is not producing Sonic Hedgehog. So if there was any Sonic Hedgehog, it was initially there, and that's something I should also mention. We initiate the system with a spike of Sonic Hedgehog at the very ventral side. But if this degrades, there is no Sonic Hedgehog. And some of the successful solutions, we call them insensitive, they still had the same size of the floor plate. And some of the solutions, we call them sensitive to Sonic Hedgehog, are completely lost the floor plate. So this is this plot showing you, the change in the floor plate size, the length of the floor plate, and there were some things in between. But focusing on those two, so insensitive, they don't, even if you remove the Sonic Hedgehog, the right from the floor plate, you still get floor plate. And if this is sensitive, you completely lose the floor plate. So now we ask, how this different distinction between classes of solution, mechanisms of floor plate formation, how they affect. Can you see a difference in the parameters that are defining these classes? And indeed, there are, in many cases, this is similar, but there are some like uniform activation, and this makes sense. Like you probably, in the insensitive class, want to have a system that activates the floor plate independent of its own Sonic Hedgehog production, and this is by this uniform activation of the floor plate. Yeah.
Do you get this distribution by averaging over many realizations of the other parameters?
Sorry, that was this, okay, and this is the full screen. So this is I didn't say that. This is for all successful solutions now divided into two classes. And now if we look into how the floor plate size is changing as the tissue is growing, and now we also perturb growth. So we wanted to see, depending on the level of growth of the tissue, with 5 being the growth that is observed in the neural tube. Now how the size of the floor plate is affected by the tissue length, and what we see is initially is its form, and then it scales with the size, and it's for insensitive, it's perfect scaling. For sensitive, not close to that. Now I will speed up a bit just to get to the end of the story. If you now plot it so that was in the absolute scale, so micrometers here. If you plot in the relative position, the tissue is growing, so you really see that the growth you have like perfect scaling, and here some for growing tissues there is some additional effect towards later times. And now if you delete this Sonic Hedgehog that is produced by the floor plate, so again you remove one of the links, you see that here nothing changes also in this scaling, and here you lose the floor plate. It's formed initially, but then because there is no this feedback of Sonic Hedgehog production, it cannot maintain itself. And we were also interested in what decides about this initial the timings. So what sets this that the floor plate is formed early on within roughly 10 hours, and one answer is it's decided partly by the parameters of the network. That also decides partly by the parameters of the network. But if you would like to name one thing that is, it's degradation, that is the one parameter that if you perturb that, you really could affect very clearly this the time of formation. So it's maybe also not surprising that you kind of take the inhibitor of the floor plate, and if it degrades faster, you could actually allow floor plate to grow quicker. And now, as for the size, so because initially I showed you also, that we have distribution of floor plate sizes for a given network. You just have one floor plate size, but depending on the parameters, you can have different floor plate sizes. This is decided by the strength of the interactions, but it's also decided, and this is I would say when the notohord comes into the story by the level of initial Sonic Hedgehog. So if you have more initial Sonic Hedgehog, and this is something like we model by adding the flux of Sonic Hedgehog to the system, you could really change the relative size of the floor plate. And here there are like large floor plates. It means these are the parameter sets that produce you floor plates that are just for a given flux. They are just larger, saying like here 20%, and here even 30 here, and 5% here for the same level of initial flux. So the level of flux matters, and it also matters when you think. That's actually the next transparency. But here, what I plot is so far I was discussing the floor plate size, and here I get to the amplitude of Sonic Hedgehog. So you have the floor plate, but you probably would like to know what would be the amplitude of Sonic Hedgehog produced by this floor plate. And here as a function of growth, it also, I would say, scales. So probably you would say, okay, maybe this is related to the floor plate size, and indeed it's related, saturates later on. But maybe here this is not the key observation. Like the key observation is that depending on the growth, this could really change. So for the floor plate size, it wasn't the case. You saw those curves perfectly aligning. For the amplitude, it's really related with the growth, but it's also related with this initial flux. And here, I would say you could see how the floor plate is actually contributing to the amplitude, saying this shows you that having the flux from the notohord, and it's like continuously provided to the system, and also having the floor plate that is producing Sonic Hedgehog, you have kind of two sources of Sonic Hedgehog in the system. And now, if you remove the flux, leave the floor plate, then if this flux was high, then you have a drop in the amplitude. And this is Sonic Hedgehog that remains, and this is Sonic Hedgehog produced by the floor plate. Whereas if this Sonic Hedgehog was, if this flux was low, probably removal of the notohord, and what we know in the development of the spinal cord is that the notohord later on moves away from the spinal cord. So, it's not quantified well, but the assumption is that the flux is largely reduced, and that's why you also need the floor plate to be there. And finally, we tested this prediction experimentally. And the idea of the experiment was first, so there are a few experiments. One of them was, this is the wild type. Here you see in red, you see the Sonic Hedgehog. In green, you see floor plate. Blue marks for the cell nuclei. And this is the wild type. So you see, if this is the Sonic Hedgehog produced by the floor plate, by the notohord. If you delete Sonic Hedgehog from the floor plate at, I think that's 7.5 E 7.5, you still have the floor plate. It doesn't produce Sonic Hedgehog, and if you quantify, this is what you see. So this points out to this insensitive class of solutions, saying it's the same but done experimentally. So in the simulation, I remove a link. In experiment, we just remove Sonic Hedgehog production from the floor plate. If you do it a bit earlier, so E 5.5 to 6.5, you delete it both from the notohord and from the future floor plate. So basically, there is no notohord, there is no floor plate, so there is no Sonic Hedgehog. Yeah, it's just empty. That's also shows that this initial Sonic Hedgehog is important in the system. And finally, if you do this kind of deletion later on, saying, yeah, that's E8.5. Yeah, again, you don't have Sonic Hedgehog in the notable, you don't have it in the floor plate, but the floor plate is still fine. Okay, so I think I will now just quickly go to the last part where I want to discuss the general properties that the system of morpho or of growing sources, growing sources. Yeah, like let me change to here. You know, it's all described in this paper, but just to give you intuition, you have now a source on one side of the domain and the source on the other side. They are producing the morphogens. They activate each other. Here, this psi stands for the concentration of the morphogens. This is diffusion. This is degradation, and this is, this is some activation through the theta heaviside function, so like a step-like function. And this guy Fi marks for the gene interactions in the linear fashion, where epsilons, if they are positive, this means activation. If they are negative, this means inhibition. The C is the threshold of activation that is additionally imposed by larger network that this motif can be embedded. And we try to stay here as general as possible, saying like the CI can be either positive or negative. And just ask a question: under what conditions on these parameters the system will result in stable pattern? Just you get two genes, two reaction, diffusion reaction equations that are coupled with interacting genes, and yeah, we would like to know whether this set of equations will result in stable pattern. And we do it, and basically Maciej Majka, a postdoc in my group, he solved it analytically. He found some conservation law. He could apply Green's function and do some nice integrals. That's fun. But by the end of the day, you want to know whether. So that's the example of one of the stable solutions, that you get two domains that hit each other, and they stay and they overlap a bit. This is delta X, and I consider this as a proxy for a region where you have a mixture of cells, so something like a proxy for patterning precision. They could also stay away from each other, these domains, and then there is a gap where neither one gene or the other is expressed. If you want to see a bit of dynamics, it's like, again, the blue domain is starting on here on the left, the red is starting on the right, and they approach each other. Like if you have a source, actively producing the morphogens that is also activating its expression, then you expect that this domain will expand. But if you have two expanding domains that are, they can inhibit each other or activate each other, something happens when they hit. We don't model the hitting as such, but we model what happens later. And what can happen is you can either have what we call indeterminate gene expression pattern, where they just go through each other, they don't care. They could also hit each other, and one would, and they would move in a kind of fashion with a constant velocity in one direction, or they could make a stable. They could hit each other and stay there. And again, that's like, probably two more slides on that. What we found is that this intermediate indeterminate gene expression profiles, they're part of the space, and the space is defined here through some, reduced parameters. But what you should take out of that is that really the stable gene expression pattern, they are very rare, but that should maybe rise an alarm, like ring a bell, like. But okay, but in development you really had a lot of these stable patterns. So what's is this model maybe missing something? The thing is, this traveling gene expression pattern close to this region of stable gene expression pattern, they travel at very low speeds. So we believe, and we also that this broad plateau, low velocity drift, is something that patterns in biology can persist. And one example we heard over these last days a lot about Drosophila. In Drosophila, in the nuclear cycle 13, the posterior genes, so this giant and NIRBs, they shift. And if you do the map, you find that they shift with this 24 micrometers per hour. They do it for a very short period, but still that would fit somewhere here. So I would say if that's one of the take-home messages from this work, that would be it. If you are more into network motifs, then we also analyzed and categorized different motifs, how they provide the stability. And finally, we moved with that we teamed up with a group of Thomas Sokołowski and his collaborators, Peter Reintenbolde and Nils Becker. They had a model with very realistic model of what happens in Drosophila, including noise and so on. But their question was about what happens if you remove the morphogen gradients, or in this case the maternal gradients, and you let the genes as such. And then they had different. The idea was that by playing with the inhibition of next nearest neighbors and nearest neighbors, you could increase stability of such a pattern of 5 stripes by orders of magnitude. And that's what simulation showed, that you get increased by roughly 2 orders of magnitude. Just this is just one plot. And then Maciej applied the theory he developed to make a prediction about what's the most stable pattern in terms of theory. You can imagine this has a lot of parameters, so he took these parameters, simplified, and then by the end of the day he got this line which was really close to that. That's no fitting here. So he could get it basically everywhere here, but it really also coincided with this peak, so we were really happy, and that's very okay. It is on archive since last year, but recently we got updated that. And to sum up, I covered I covered plenty of aspects of pattern formation. One of that is the signaling part, and in my work, and also in the works of others like Gasper, Thomas Gregor, Petkova, Erik Wischhaus, Białek, it seems like the signal is decoded in the optimal way, saying like what acts as the decoder is the gene regulator network, but at the end you get something that really uses the available information very accurately, saying probably this indicates that there is some evolutionary adaptation, at least in Drosophila or spinal cord, maybe in other system that drove the system to this acting close to the optimal limit. But that's the signaling part is related to the mechanical properties of the tissue, and in this example we saw that early on the tissue is rearranging a lot. Later on it kind of gets to the solid-like stage. So it's unclear for me. I could always say the evolution make it this way, but that's like very very hand-waving argument. You probably could say there are other alternatives, and then that's one. This one is actually performing best, but I would like to say that there is still much to do there. And then that should be summary part 2. We also consider this additional feedback like in the source formation, but what you show, so the take-home message from that is it's still relatively quickly formed, and then there is scaling of the floor plate size. And finally, you could move to more theoretical framework and ask these questions. Knowing just the structure of your equations, diffusion equations, and whether there are some general properties that govern the behavior of these systems. And with that, I would like to thank people who were involved in these projects. The IST people, I spent 5 years there as a postdoc. Great time, I could recommend IST. Then I got back to Jagiellonian University, also for me, very fruitful homecoming. And then people who are now in James Briscoe, like through UK, Tobias, my first supervisor at IST, now at the University of Cologne, Thomas, my colleague from IST, and my group that we often have lots of discussion. These faces are changing, but we still stay in touch. And just if someone, there are some undergrads here who wants to come to Krakow, have a PhD, we also have additional PhD funding apart from the regular scheme. I could offer, in this project, additional scholarship. So thank you for your attention.
Thank you so much. Any questions?
So, in your error calculation for boundary specification, which you discussed in the first part of the talk, and then I think you also discussed this in your earlier part 1.
Okay. You mean... sorry.
Where you calculate information and...
Okay. So that's mostly this pedagogical talk, but here it was... sorry for clicking. Let me do it differently. This is...
So for these calculations, do you take into account of cell-cell interaction?
No. It's well, the short answer is no, because there is no cells there even, and this is that's one of the things maybe tomorrow in the tutorial I would like to discuss. That basically the discreteness of the tissue might affect this. What is neglected here is the covariance or correlation between the signals. But what we showed is that in this system, this is negligible. Saying either you include this, we have this from measurements, or you exclude that, it doesn't affect. However, recently, and I think recently this year, there was paper by I think Thomas Gregor, Will Białek was on that. The last bits of positional information, where they show that in Drosophila, before we had this small discrepancy between input and output, and now they were able to quantify that by adding this additional 0.2 bits, I think, simply by taking into account the correlation in the noise, providing, I think, I'm not sure it's still probably appropriate, but providing the first evidence that the noise as such might be also used to decode or to increase the level of information.
So one last question. So most of the data that you've shown here today, and also in your earlier talk, it's from chick embryo. Am I right?
No. So this is from mouse embryo. I forgot to. If I forgot now, this is the mouse embryo, with this exception that if we do some of the explants, saying cut part of the tissue, like for this decoding map, because there was a lot of conditions to reconstruct that. That was from the cheek explant.
Yes.
But but the cheek and mouse, to some extent, they are very similar. And if you want to know more, there's a paper by Kicheva, like a first author, Science 2014, where she studies how number of neural progenitors changes, and this is done both in mice and both in cheek.
So this BMP and sonic hedgehog signaling, this is sort of conserved in all of the vertebrates. You can.
To my knowledge, in most, yes, but again, I know that in some, I think zebrafish is reverted, saying like, okay, I'm not the biologist, so I don't make speculations, but yeah, it's at least to my knowledge, sonic hedgehog is conserved. Yeah,
Thanks.
So I didn't get the part where you're saying that the feedback is required, like the output signal from the information decoder is fed back to it's controlling the input signal, which is the morphosync. So why is it required? I mean, what is the purpose of that?
Very good question. Like, I would say it's to some extent. So that was our working hypothesis. But later on, we showed, and I should mention that when you remove the production of sonic hedgehog by the floor plate, you basically directly remove this feedback. Okay, and you show. So this is this insensitive class that it doesn't affect the floor plate size. So but still, if anything, it affects the amplitude. So let's say of sonic hedgehog in the system. So I wouldn't say this feedback is really strong there. Kind of you start to consider many options, but by the end of the day, you end up with a solution that shows you if you would found that the class of the solutions that is consistent with the experiment is the sensitive class of solutions, the ones that really require feedback. Saying like,
what is this feedback? Is it
okay? When I say feedback here, okay, here at this plot, this is just a way of plotting effectively some. If you would say you have, okay, maybe more formally, if you have these equations here, so this is this is your signal, sonic hedgehog. This is your output, floor plate, and end, so target domain. If you would have just this, and that's what we had in the spinal cord previously, there's no like, okay, direct influence of that, or this would be like an input. There's nothing that regulates sonic hedgehog, but here, the sonic hedgehog is produced by, the level of its production is decided by F. So if you would like to, interpret this in the feedback in mathematical sense, this gives you, if I would forget about this diffusion, sonic hedgehog is like, yeah, it's really on the top, nothing affects that in the dynamics of the system. However, here, F could potentially, no, not potentially, it's directly affecting the production of sonic hedgehog. So that's the feedback. And now you know how important this is.
Yeah.
This is different question, and this is what we wanted to answer with this whole scheme. And I would say, yeah, the answer is, the answer is, I would say, a bit complicated, because you would say you certainly need floor plate to be there to have sonic hedgehog, but later on, if you remove this term, because KFS means basically zeroing this term, you remove the feedback, the system is still fine. Okay, in the sense that it's fine in terms of having the size of the floor plate of the same magnitude, but of course the amplitude of sonic hedgehog will go down. So in these terms, I would say you need sonic hedgehog for development, especially you need it early on. Later on, and that's also my previous work, we showed that if you switch off the sonic hedgehog 10 hours or 20 hours after, establishing a pattern. Not after establishing a pattern. That's even before the stripes are formed, but you just remove signaling. You just need the signal for 10, 20 hours. Then it's still fine. Then the pattern is reproduced with 90%. So yeah, that's there are interesting connections here.